My name is Shabana. I'm a Ph.D. candidate, and I'm working with Dr. Mark Paschke, who's a professor in the Forest and Rangeland Stewardship Department. So I'm basically trying to look at arbuscular mycorrhizae communities and how they will influence restoration ecology in different types of grasslands, including the shortgrass steppe and tallgrass prairie. So mycorrhizae is a microscopic fungus, and it makes an association with about 80% of land plants, so it's found everywhere. But it's a fungus in which the plant benefits and the fungus benefits, so it's a symbiotic relationship. The best way to identify arbuscular mycorrhizae is by high-throughput sequencing. When we extract the DNA using our soil kits, we're only using about 0.5 grams of soil. So it's a really, really small amount, but if we want to see what's in that soil, we have to amplify it a lot. One plant species can have between like 10 to 20 different mycorrhizal species associated with it. Some of my questions started from, if you have so many species in network, what makes those species work well together and what makes them not work well together? The first chapter of my dissertation looks at a field study and a greenhouse study, in which the field study is in Nebraska, where we took soil samples looking at different levels of plant community diversity. What is nice about that is that I was able to recreate that same kind of plant community in the greenhouse using soil samples from the field to inoculate the pots that I was growing in the greenhouse. With both projects, in the field and in the greenhouse, we're trying to look at how different levels of plant community diversity might be influencing the structure of the soil microbial community. Next steps are to create a disturbance. In the field study, the disturbance that we're going to be looking at is rototilling. And in the greenhouse study, the disturbance that we'll be looking at is literally just chopping off the above ground, so all the plants. We might be able to understand from our studies how pieces of a disturbance are influencing the soil microbial community. For the last chapter, what we're planning on doing is planting a completely different seed mix, something that maybe includes a lot of grasses and few forbs, which I think is typical in a lot of restoration settings. And so we're going to replant in the greenhouse pots a less diverse plant community, and then see how that then, after the disturbance, changes the soil microbial community. Essentially, down the line, I'd like to make it easier for land managers and for restoration ecologists and for people that are trying to apply mycorrhizae, trying to make it so that we can understand some basic fundamental questions about mycorrhizae and its ecology in order to better apply and better utilize it in restoration settings and potentially, all sorts of settings, really, all sorts of ecosystems.